So in this video, let me run through the test review for chapter 16 on proteins and enzymes. One thing you should be able to do is to write a complete synthesis reaction for a given di or tri or tetrapeptide. Now here we see the 3-letter abbreviation. You should also be able to use the single letter abbreviation for these amino acids. Like for example, for this tripeptide, instead of writing out the 3-letter abbreviation, this tripeptide could have also been abbreviated using the single letters, which would be CSK. The L is, I think, given to leucine, and so lysine has K as abbreviation. One other aspect to this is to know that when we see a sequence of amino acids such as these, the first one is always talking about the N terminus N, or the amino N, and then the right-hand side would be the carboxylate. And actually, we're going to be writing the ammonium structure for this one. So if you're doing the synthesis reaction, well, first thing you're going to write the 3 individual amino acids, and we're going to use the Zwitter ion because that's the most common form of it. And remember, for the Zwitter ion, both of the functional groups on the ends are ionized, right? It's this core feature that shows up on all the amino acids. There's the alpha carbon. The alpha carbon connects the amino group, or I should say the ammonium group, with the carboxylate group with the R group, or side chain, however you want to call this. The first one is cysteine. That's our thiol. There we go. Now, when you're laying out the amino acids on the reactant side, it really doesn't matter which order you put it in. Usually I'll put it in as I see them. There's the common features again. Serine is our smallest alcohol. And then lysine. Lysine's one of the charged amino acids. So it's not only charged when it comes to our ammonium group here and the carboxylate group here, well, also the side chain. And the lysine has the ammonium group at the top. And I'm going to condense this. There's actually 4 carbons, 4 CH2 groups, and then the ammonium group. So it has a charged R group here. Okay, so this is a condensation reaction, and so water is going to form. We'll get to that in a minute. And basically what's going to happen is that each place here, you're going to get a peptide bond. And while I'm thinking about it, every time a peptide bond forms, you're going to form water. So since there's 2 here, well, I'll go ahead and write it in. So 2 water molecules are going to form here. Now, when the peptide bond is formed, then the charges disappear. But everything else really just stays in the same place. R groups stay in the same place. The ammonium at the end, the carboxylate at this end, they stay charged. And it's just sort of a matter of patiently putting all the pieces together. I'm going to write the peptide bond out in more of an expanded form so we can see that. Notice it is an amide functional group. I'll make a note of that in just a second. And notice the first thing I'm doing, I'm just getting the peptide bond alpha carbon in place to sort of set up my chain there. Don't want to forget that charge. And now I can go back with the same R groups. And right now the sequence of R groups, that's what matters. That should match up with the sequence of amino acids that's laid out in the notation there. And that's it. And don't worry, I'm circling the charges. You don't have to. Actually, I'm not even circling all of them, am I? Doesn't matter. So this problem is done. Let me just make one other comment just to remind us. Right, when we're talking about a peptide bond, these are amide functional groups. Now the next question is dealing with denaturation and inhibitors. Actually, let's do this one because this we can pretty much knock the whole thing out of in one statement. This one's a little bit more involved, the top one there. And we want just a couple of items to distinguish, in this case, competitive inhibition versus non-competitive inhibition. And if we write a few more than 2, that's fine. So one thing about the competitive inhibition is that the inhibitor, which I'm just going to abbreviate I here, for the competitive inhibitor, it binds in the active site. So really what it's doing is competing with the substrate. And let me abbreviate that S. And oftentimes, since it's binding in the same location that the substrate binds, well, the inhibitor is going to look like the substrate, at least on the end that binds in the pocket, binds in the active site. And I should probably put quotation marks around look like. It's going to have the same chemical signature as the substrate. Right, there's just going to be a small portion of the inhibitor that has a similar chemical signal or signature as the substrate, so it can sit in that little pocket that we call the active site. Non-competitive inhibition, well, the inhibitor will say bind somewhere else on the enzyme. And we can kind of, there's sort of a complementary statement for each one of these. So the inhibitor does not compete with the substrate. And then also, the inhibitor does not look like the substrate. In both cases, the inhibitor is going to shut down the enzyme or slow down the enzyme. It can slow down the reaction. Actually, the way, the inhibitor here is going to block the active site so the substrate can't sit into it. On this one, the inhibitor changes the shape of the enzyme. And so by altering the shape, then it alters the shape of the active site, prevents the substrate from sitting in the active site. And I'm sure there's slight variations in the theme here. I'm not a enzyme chemist, so these are just sort of the basic ideas with the two different types of inhibition. Now for denaturation, there's actually 5 different types of denaturation that we talked about in chapter 16. And we're just looking at the change of pH versus organic solvents. So for acids and bases, well, what is an acid-base reaction? Well, it's like proton transfer, and what the proton transfer is going to do is it's going to interfere with two main tertiary interactions on the protein, and so those are going to be salt bridges and hydrogen bonds. Now, the salt bridges are the charged structures. Like lysine we saw just a few minutes ago, that has a plus charge, sticking out somewhere on the protein. And so it could bind with something like glutamic acid, and the negative and the positive charge link together. If you throw in like an acid, it can protonate the carboxylate group on the glutamic acid, and then it kills the salt bridge interaction. And in effect, right, that's what's denaturing the protein. The organic solvent, well, its primary target is it affects hydrophobic interactions. So again, these are just 2 of the different denaturation or the denaturants it's talked about in chapter 16. You should review that material and look at the other ones and be able to say the same thing, like agitation and heating will affect also affect the hydrogen bonds, sort of some of the weaker interactions within the structure, within the protein structure. Another one is like metal ions will denature the protein by attacking the disulfide bonds. So be familiar with those different processes there. So in the next question, you see some of those examples like a disulfide bond, etc. These are referring to the, different interactions, different descriptions of proteins. And we want to identify whether it's primary, secondary, tertiary, or quaternary. First off, let's look for the quaternary ones, because what you're always looking for in this kind of interaction is some description of a multi-subunit protein. So 5 subunits versus a single subunit versus 2 subunits. So if you have 2 or more subunits, in some ways, it doesn't matter the rest of the description. It's going to be referring to one of these quaternary interactions. Now the other 3, primary, secondary, tertiary, they're dealing with single subunit interactions. Sometimes that's stated explicitly, sometimes it's not. Like for example, for B, if you read this, hydrogen bond occurring between peptide bonds, these interaction peptide bonds, what they're describing are really alpha helices and beta sheets. This is the definition of a secondary interaction. And then for C, the main thing you're looking at is that it's actually referring to the side chain interactions. Basic acidic amino acids actually is one that I just talked about a second ago, salt bridges. So if it's side chains or R groups, remember there's 2 different terms for those things. If it's within a single subunit, that's always talking about the tertiary interactions. Now, of the ones that are listed here, it's the primary list that you don't see. Well, we saw a primary a second ago, that first problem. So the primary sequence is just the actual amino acid sequence, where there's no reference to like alpha helices or interactions or whatnot. It's just a core, what amino acid is attached to, which amino acids are attached together there. Chapter 16 discussed a little bit about how we draw amino acids. So yes, it's true, right? All amino acids have this. And I'll answer the question sort of in a roundabout way here, like I tend to do sometimes. Okay, so all amino acids have this feature, and then depending on the specific amino acid, then the R group is going to change. The valine is the one that has the isopropyl group sitting up here. So this is the Zwitter ion, right? And this molecule is the one that forms when the pH is equal to the isoelectric point. I don't expect you to memorize that list of isoelectric points. Most of them are around 6 for what it's worth. What you need to realize is that as the pH changes from the isoelectric point, what's happening to this structure? Okay, so let's sort of think our way through this process. What does it mean to be at pH 2 compared to a pH of 6? Right, because let's see, right, this pH here would be a pH of 6.0. Again, just as Zwitter ion. So if I go from a pH of 6 to a pH of 2, what I'm adding protons. I'm making the solution more acidic. How are protons going to affect the ammonium group versus the carboxylate group? Yes, it's going to bind with the carboxylate group. And so when you have a pH that's lower than the isoelectric point, the carboxylate group becomes protonated, and then the overall charge of the amino acid is a +1 charge. And so now the actual answer to this question is going to be in green. Again, all of this is the same. Oh, that's not the same. Right? This group is going to be protonated. This ammonium group is not affected. If anything, excess protons, is going to allow this to stick around even more. This R group, since it's a non-ionic R group, it's not affected. And so this is the structure. And again, overall has a positive charge now. Overall charge here is 0 for this amino acid. We talked about the stereochemistry of the amino acids, and the amino acids that we see in nature are the L amino acids. And we really focused on the Fischer projection when we wanted to illustrate that. Alanine has an R group, one of the simpler ones. It's just a methyl group. Now, this is not the Fischer projection. Okay, remember what the Fischer projection is. It is essentially the longest carbon chain written vertically on the page. And for all of the amino acids, it's the carboxylate group that's going to show up top. The alpha carbon, 19 out of the 20 amino acids. The alpha carbon is chiral. The only one that's not chiral is glycine. And then down here at the bottom is the R group. And then what's left? Well, what's left is an ammonium group and a hydrogen. L, left-hand side. Left-hand side. This is where the ammonium group is going to be, and then the hydrogen is going to be on the right-hand side. So 19 of the 20. So I guess technically glycine wouldn't have a Fischer projection because there's no chiral carbon. On all the others, it would look like this. Only thing you're changing is the R group down here on the bottom end. And the last question that we see dealing with chapter 16 is the number 10 here. So what you have to imagine is these little pigtails. These are, imagine an alpha helix. In the middle are R groups. And what I want you to be able to do is identify the type of, you can see it in the language here, non-covalent interaction between these R groups. What's being described here? So these R groups, this would be valine, this would be alanine. You can imagine it's sitting there on the alpha helix somehow, and the R group is poking into the interior portion of the protein. Right? Like dissolves like, nonpolar interacts with nonpolar. The specific term in protein chemistry for this type of interaction is hydrophobic. Why would a nonpolar R group crowd on the interior portion of a protein? Well, because water is surrounding this protein, and nonpolar groups are hydrophobic. They don't like water. So it doesn't make sense for these hydrophobic units to be sticking out into the aqueous environment around the protein. Instead, they're going to crowd together with themselves in the interior portion. Now, on this end of things, what we're looking at, notice there's an ion here, so there's a charged state. This is serine. This is not a charged state. This is just an alcohol, but it is polar. The carboxylate, or the carbonyl group of this guy, is also polar. And what do we have when we have an OH group interacting with a polar bond on another molecule? Well, this interaction, and we've seen it many times in this class, this is referred to as a hydrogen bond. So these are 2 of the 5 that are mentioned in this chapter about the tertiary interactions. And I won't write everything down here because it'll get kind of messy, but take a look back at the notes, review a video. Actually, I will scribble in, let's say 1, 2. This numbering is probably different than what's in the video, okay, or in your notes. Another one is a salt bridge. That's between ionic R groups. Another one is a hydrophilic interaction. This one is a polar or charged amino acids on the exterior portion of the protein interacting with the water that's surrounding it. And then the 5th one here would be a disulfide bond. And that would be between 2 cysteine amino acids. That's the only one. Actually, that one's an actual covalent interaction. Let's just sort of make note of that. So it's pretty strong. Right? There's a physical bond. It would look something like, well, just to save room or make room, that's this 2 sulfur atoms bonded together. That's a disulfide bond. So of the 5, this is actually a covalent interaction. These other 4 are non-covalent. And that does it for the test review for Chapter 16. Thanks.